Hello, my name is Olivia Barrow, and I decided to do my presentation on Liddle syndrome. So, what is Liddle syndrome? Liddle syndrome involves a protein, alpha epithelial sodium channel, and the mutation through the cysteine number 479, which is replaced by arginine. The condition which results from this mutation is Liddle syndrome, genetically inherited high blood pressure, which I will be referring to as hypertension. Epithelial cells serve as another barrier of protection for the body against foreign objects and viruses. Epithelial sodium-potassium channels play a major role in the management of extracellular fluids that contain electrolytes used by the body. In these regulatory proteins, sodium ions flow from the extracellular fluids into the cytoplasm. The sodium is processed and passed back out into the fluid through the sodium-potassium pumps. I decided that these proteins are regulation proteins because they are responsible for the sodium/electrolytes being used in animals. They are responsible for the homeostasis of the body and aid with the maintenance of energy. Changes to the protein due to the mutation, so the protein becomes more active because the mutation causes a change in the signaling region of the subunit used for degradation to occur. As a result, more channels appear on the exterior of the cells, and the subunits are not degraded. With more channels, the reabsorption of sodium occurs, which leads to hypertension and the removal of potassium from the bloodstream. The proteins begin to work overtime and take too much away from the system at one time, causing the system to fail. The location of the proteins: the alpha epithelial sodium channel is located on the apex membrane of epithelial cells located in the lungs, respiratory tract, kidneys, reproductive system, and other organs in animals. So to show my 3D model, I'll be showing the normal and the mutated protein. So the beta is represented by green, and the alpha I use red felt, and then for gamma I use blue felt to show the different channels on each side. The model does not allow us to see how the molecules can flow through the channel. It's kind of tight, and all we see in the middle is, I left a little bit of room, but you can't really see how these channels can be used. You kind of see it from the front, how side by side you would have the cell next to it, and it would be on the outside of the cell, the exterior. And it doesn't really show us how information is exchanged. It's kind of like just an overview, just a little model. It doesn't really show the function. So to show the mutation, which is what I have in front of me, I utilized some glittery balls and glittery string, and I attached it to the tubes, and I put the balls on top to show the mutation. So through this second model that I use, it doesn't reflect a change in the number of channels that begin to appear on the cell. And it doesn't also show how the system has begun to work overtime and do its processes at a faster rate than before. It's kind of just showing how the structure has just been mutated. So changes to the structure from the protein. So the main functional group that's involved in the mutation and just in epithelial channels is the amino functional group. So some properties of that side chain. The exterior of the channel is highly empathetic with hydrophilic pores within the structure to contribute to the intake and release of water. The protein is larger compared to other proteins due to the functional properties and operations of the channels. It must take up more surface area in the cell's exterior to perform the proper maintenance of homeostasis. So the bonds of the original side chain, the bond of the original AA is a peptide bond. Most proteins are composed of amino acids held together by peptide bonds. During the reaction, water is released, which is an essential part of the epithelial sodium channel process, dehydration. So some properties of the new mutated side chain and the bond involved. So the new mutated AA side chain is much more hydrophilic than the normal side chain due to the alteration of the amino acids that the mutation caused. The protein becomes more salt sensitive and begins to grow in size and numbers. The main bond involved in altering the shape of the protein is disulfide bonds. Disulfide bonds reverse the natural reaction by utilizing the components of a covalent bond. Usually proteins that are under oxidative stress create these bonds. Amino acids are still involved in the formation of the protein, but due to inflammatory conditions within the cell, cysteine residue is created. Through the use of cysteine residue by oxidative folding, the new bonds are created. And finally, the mutation's effect on the protein. So the mutation causes the protein to overwork itself. Sodium is absorbed more rapidly by the collecting tubules, which causes the lumen to be electronegative, which means less potassium is released. In the organism, a number of symptoms can occur due to changes, including hypertension, hypokalemia, and metabolic alkalosis. So that is my presentation on the syndrome, and thank you.